The year was 1538, and in the damp stone chambers beneath the Palazzo Vecchio in Florence, a man named Giovanni Battista Ramusio bent over a peculiar manuscript illuminated with creatures so fantastical that even he, a seasoned collector of travellers' tales, found his breath catching in his throat. The parchment had arrived from the Venetian merchant galleys that plied the spice routes, and it depicted what the Portuguese sailors called peixe mulher, the woman fish, which they swore upon their immortal souls they had witnessed in the waters off the Malabar coast. Ramusio's trembling fingers traced the marginalia where an anonymous scribe had noted in cramped Latin that the creature's song had driven 3 men to leap from the ship's deck into the moonlit waves, never to surface again. What Ramusio could not have known, sitting there at the gathering Tuscan twilight, was that the manuscript described not a mermaid, but rather a phenomenon that would baffle natural philosophers for 4 centuries hence: the complex vocalizations of humpback whales, whose haunting songs can travel across entire ocean basins, penetrating depths where sunlight has never ventured and will never go. The ocean kept its secrets with a jealousy that would have impressed the most paranoid Medici prince, and in those years when men still believed the world might end at some unknown edge, the creatures of the deep existed in a realm between natural history and pure mythology. In the markets of Lisbon, where the smell of salt and dried cod mingled with the incense from 100 churches, fishmongers displayed the carcass of what they called a sea monk, its body vaguely humanoid beneath the layers of decomposition and imaginative taxidermy. The Holy Office examined it with both ecclesiastical concern and morbid fascination, debating whether such a creature possessed a soul and whether its existence challenged the divine order or confirmed it. What they were actually examining was likely an angel shark or some species of ray, its cartilaginous structure collapsed and distorted by days in the Mediterranean sun. But the classification of marine life in that era depended less on anatomical observation than on theological interpretation, and the unreliable testimony of sailors who had spent too many months at sea with nothing but hardtack and watered wine for sustenance. Yet even then, scattered throughout the ports and monastery libraries of Europe, there existed fragments of genuine knowledge, small jewels of observation embedded in the mud of superstition. In the year 1551, a French naturalist named Pierre Bellon published a work in which he noted that dolphins, despite their fish-like form, breathed air and nursed their young with milk, a fact that placed them in a category that confounded Aristotelian taxonomy. Bellon had dissected a dolphin that fishermen had brought him from the Bay of Biscay, and he observed with meticulous care that its internal organs bore more resemblance to those of a pig than to those of a mackerel. This observation earned him both praise and condemnation, for it suggested a fluidity in God's creation that some found uncomfortable. The dolphin's brain, Bellon noted, was proportionally large and convoluted, though he could assign no function to these convolutions beyond the general consensus that the brain was primarily an organ for cooling the blood. The truth that dolphins possess cognitive abilities that would astound these early observers, that they can recognize themselves in mirrors, mourn their dead, and communicate with signature whistles unique to each individual, lay beyond the conceptual horizon of 16th century natural philosophy. across the ocean that Bellon's dolphins called home, in the waters of the Caribbean, where Spanish galleons fought a losing battle against both English privateers and the wood-boring teredo worms that could reduce a ship's hull to Swiss cheese within a season, there lived a creature that the Taino people had hunted for generations with a reverence that the conquistadors mistook for primitive superstition. The manatee, which the Spanish called vaca marina, grazed on seagrass meadows in shallow coastal waters, its massive body moving with an improbable grace through the crystalline depths. A fully grown manatee could exceed 1,300 pounds, its weight sustained by an endless consumption of aquatic vegetation, up to 15% of its body weight daily, a quantity that required the animal to spend most of its waking hours in a meditative state of perpetual eating. The Taino had understood something that would take European science centuries to articulate, that these gentle creatures, with their whiskered faces and paddle-like flippers, were architects of their ecosystem, their grazing patterns maintaining the health of seagrass beds that in turn provided nurseries for countless species of fish that fed the communities along the shore. When Christopher Columbus reported seeing mermaids in these waters and noted disappointedly that they were not as beautiful as painters had led him to believe, he was almost certainly observing manatees, their rotund forms distorted by distance and the peculiar optical properties of water-meeting air at the surface of the sea. The anatomical peculiarities of marine creatures presented puzzle after puzzle to the natural philosophers who began, in the 17th century, to approach the study of life with something resembling systematic methodology. In Amsterdam, where the wealth of an emerging empire funded both art and science with equal enthusiasm, anatomists obtained specimens from the Dutch East India Company's ships, creatures preserved in barrels of gin and rum, their flesh pickled but their structures still discernible under the knife. One such specimen was a nautilus, its shell a perfect logarithmic spiral that seemed to encode mathematical principles that human minds were only beginning to articulate. When sectioned, the shell revealed chambers, each one slightly larger than the last, separated by walls of such delicate mother-of-pearl that they seemed too fragile to withstand the pressures of even shallow water. Yet these chambers served a function of elegant efficiency. By adjusting the ratio of gas to liquid within them, the Nautilus could control its buoyancy with a precision that allowed it to migrate vertically through the water column, rising at night to hunt and descending during daylight hours to depths where predators were less common. The Nautilus, it would eventually be understood, was a survivor of ancient lineages, its basic body plan unchanged for hundreds of millions of years, a living fossil that had witnessed the rise and fall of the dinosaurs from its position in the Paleozoic seas. But in those early years of comparative anatomy, before the concept of deep time had shattered the comfortable chronology of biblical literalism, the Nautilus was simply a wonder, and wonders were collected by princes and scholars with the same acquisitive passion they applied to rare books and religious relics. The cabinet of curiosities assembled by Frederick Reich in Amsterdam included specimens that blurred the line between scientific preparation and artistic creation. He preserved sea creatures in liquids of his own formulation, the recipes for which he guarded as jealously as any alchemist guarded the formula for the philosopher's stone. His preserved octopi sprawled in glass vessels, their 8 arms frozen in gestures that seemed almost purposeful, each arm lined with suckers that, under magnification, revealed structures of astonishing complexity. Each sucker was a marvel of biological engineering, capable of tasting and smelling as well as gripping, providing the octopus with sensory information distributed throughout its body rather than centralized in a single brain. This distributed intelligence, which allowed an octopus to solve problems with one arm while the other 7 pursued different tasks, suggested a mode of consciousness so alien to human experience that even today neuroscientists struggle to find adequate models to describe it. The 17th century also brought the first serious attempts to plumb the depths of the ocean itself, motivated less by pure scientific curiosity than by the practical concerns of naval warfare and commercial shipping. In England, where the Royal Navy was becoming the instrument of imperial ambition, there arose a need to understand the behaviour of cannonballs as they arced through the air and the trajectories of ships as they navigated currents that could add weeks to a voyage or subtract them. Robert Boyle, whose work on the properties of gases would establish fundamental principles of physics, became interested in the question of pressure at depth, hypothesizing correctly that the weight of water above must compress anything submerged beneath it. He conducted experiments with sealed containers lowered on ropes into the Atlantic, retrieving them crushed and deformed, their brass fittings buckled by forces that increased with every fathom of descent. What Boyle could not observe directly was how the creatures of the deep had evolved to not merely survive, but thrive under pressures that would instantly kill any surface-dwelling animal. The bodies of deep-sea fish contained no air spaces to compress. Their tissues were suffused with water, making them effectively incompressible, while specialized proteins kept their cellular machinery functioning in the cold and crushing dark. Some species developed swim bladders filled with lipids rather than gas, maintaining neutral buoyancy without creating pockets of compressible air that would collapse in the depths. Among the specimens that occasionally surfaced in fishing nets or washed ashore after storms, few captured the imagination quite like the giant squid, though for most of history it existed more as rumour than as confirmed reality. Norwegian fishermen spoke of the kraken, a creature large enough to drag a ship to the bottom, its tentacles capable of plucking sailors from the rigging like a child picking berries from a bush. These accounts were dismissed by natural historians as the exaggerations of simple men whose diet of fish and aquavit had addled their perceptions. Yet the occasional piece of physical evidence, a tentacle the thickness of a man's torso, lined with suckers bearing serrated rings of chitin, suggested that something of enormous proportions did indeed inhabit the ocean's depths. The giant squid, Architeuthis dux, would not be scientifically described until 1857, when a Danish naturalist examined a specimen stranded on the coast of Jutland. But even then the creature's behaviour, habitat and life cycle remained almost entirely mysterious. It was not until the late 20th century that scientists obtained footage of a living giant squid in its natural habitat, revealing a creature that moved through the mid-water depths, with tentacles extended like fishing lines, waiting for prey to blunder into reach. The largest confirmed specimen measured nearly 43 feet in length, though whalers occasionally found circular scars on sperm whales that suggested even larger individuals existed in the uncharted deeps. The relationship between sperm whales and giant squid represented one of the ocean's great predator-prey dynamics, an ongoing evolutionary arms race conducted in absolute darkness at depths where the pressure would crush a human body into jelly. Sperm whales, the largest toothed predators ever to exist on Earth, dove to depths exceeding 7,000 feet, holding their breath for over an hour as they hunted in realms where no sunlight had penetrated for billions of years. Their echolocation clicks, produced in a specialized organ in their massive heads, were the loudest sounds produced by any animal, capable of stunning or even killing prey with focused beams of sound energy. The spermaceti organ that gave these whales their name, a massive reservoir of waxy oil that could constitute a third of the animal's total length, served multiple functions. It focused and directed the biosonar clicks, and it controlled buoyancy by altering the temperature and thus the density of the spermaceti through a complex system of blood vessels that could be constricted or dilated at will. When 19th century whalers slaughtered sperm whales by the thousands to render this oil into candles and lamp fuel, they were literally illuminating their parlours and streets with the biological machinery that allowed the greatest predator in the sea to hunt in utter blackness. The whaling industry of the 18th and 19th centuries, brutal and efficient in equal measure, produced an unexpected dividend for natural science, an abundance of anatomical material that allowed researchers to study cetacean biology in detail that would otherwise have been impossible. In the processing stations of Nantucket and New Bedford, where the reek of rendered blubber hung over the docks like a tangible presence, naturalists picked through the viscera of slaughtered whales, documenting organ systems that defied terrestrial comparison. The heart of a blue whale, the largest animal ever to exist, weighed over 400 pounds and beat only 5 times per minute when the whale dove deep, conserving oxygen through a bradycardia so extreme that it would kill a human within minutes. Yet that same heart could accelerate to 37 beats per minute when the whale surfaced, oxygenating blood at a rate sufficient to sustain an animal over 100 feet long and 200 tons in weight. The arteries were large enough for a human child to crawl through, and the volume of blood they carried, 15,000 litres or more, would fill a small swimming pool. This massive circulatory system served muscles packed with myoglobin at concentrations 10 times greater than terrestrial mammals, allowing whale muscles to store oxygen the way a camel stores water, hoarding it against the long breath-hold dives that defined their existence. But the giants of the cetacean world, impressive as they were, pursued prey that would have seemed comically small to observers unaware of the ocean's fundamental ecology. Blue whales, those leviathans capable of weighing as much as 30 elephants, fed almost exclusively on krill, shrimp-like crustaceans rarely longer than a human thumb. This paradox, the largest animal sustained by some of the smallest, reflected a fundamental truth about marine food webs that terrestrial ecosystems could not replicate. The productivity of the ocean was measured not in acres of grass but in microscopic phytoplankton, single-celled organisms that drifted in the sunlit surface waters, converting solar energy into biological tissue through photosynthesis. These phytoplankton were consumed by zooplankton, which included krill, which were in turn consumed by whales in quantities so vast that a single blue whale might eat 4 tons of krill in a day. The efficiency of this short food chain, with only 1 or 2 steps between primary producer and apex consumer, allowed the ocean to support animals of a size that would be impossible on land, where the longer food chains meant that energy was dissipated across more steps, leaving insufficient resources to sustain truly gigantic herbivores. The krill themselves, often dismissed as mere whale food, possessed biological adaptations that would have fascinated Ballon or Boyle had they known of them. Antarctic krill, Euphausia superba, gathered in swarms so dense that they coloured the ocean pink and could be detected by satellite imagery from space. Aggregations containing millions of tons of biomass pulsing through the Southern Ocean like a single organism. Individual krill possessed photophores, light-producing organs scattered across their bodies that they used for communication and possibly for camouflage, matching the downwelling sunlight from above to make themselves less visible to predators attacking from below. They could live for up to 10 years, unusual longevity for such a small creature, and during the lean Antarctic winter when phytoplankton blooms ceased, they could actually shrink, metabolizing their own body mass to survive until spring brought renewed abundance. This capacity for degrowth, for reversing the usually inexorable march towards senescence, hinted at cellular mechanisms that biogerontologists would later study in hopes of understanding aging itself. The bioluminescence that krill employed represented just one expression of a phenomenon so widespread in the deep ocean that it constituted the norm rather than the exception. In the pelagic zone, the realm of eternal night that began around 3,000 feet beneath the surface and extended to the abyssal plains miles below, an estimated 90% of creatures produced their own light through chemical reactions that were variations on a common theme. Luciferin molecules oxidized by luciferase enzymes, releasing photons in the process. The purposes of this living light were manifold and sometimes overlapping. Some fish used photophores arranged in patterns unique to their species and sex, a visual language of identification in a realm where finding a mate might otherwise be impossible in the vast darkness. Others employed bioluminescence as a lure, most famously the anglerfish, whose females carried a modified dorsal spine tipped with a glowing escape that dangled before their cavernous mouths like a fishing pole, attracting smaller fish that mistook the light for prey, only to become prey themselves. Still other species used bioluminescence as a defense, either startling predators with sudden flashes or, in a strategy of baroque complexity, ejecting clouds of glowing mucus that clung to the attacker, marking it as a target for larger predators, while the original prey escaped into the darkness. The anglerfish represented an extreme in sexual dimorphism unmatched elsewhere in the vertebrate world, a reproductive strategy so bizarre that when first described, it was met with skepticism even among scientists accustomed to the ocean's peculiarities. Female anglerfish of certain deep-sea species could exceed 3 feet in length. Formidable predators equipped with distensible stomachs that allowed them to swallow prey larger than themselves, a necessary adaptation in an environment where the next meal might be days or weeks in coming. Males of the same species, by contrast, rarely exceeded an inch in length and were effectively parasitic, their entire existence oriented toward a single goal: finding a female in the lightless immensity of the deep ocean. When a male encountered a female, he would bite into her body, and over time his tissues would fuse with hers, their circulatory systems merging until the male was reduced to little more than a pair of gonads, permanently attached and dependent on the female's bloodstream for all nutrients. The female, for her part, gained the ability to reproduce whenever conditions were favourable, carrying her mate, or sometimes multiple males, attached like grotesque ornaments, with her always. This arrangement solved the problem of reproduction in an environment where population densities were so low that finding a mate through the conventional means of search and courtship would be nearly impossible, evolution having determined that permanent parasitism was preferable to probable celibacy. The extremophile existence of deep-sea creatures required physiological adaptations that pushed the boundaries of what biology could achieve within the constraints of chemistry and physics. The hydrothermal vents discovered in 1977 along the Galapagos Rift, a mile and a half beneath the Pacific surface, scientists found ecosystems that operated on principles fundamentally different from those governing the rest of the biosphere. Here, where superheated water rich in hydrogen sulfide and heavy metals erupted from cracks in the oceanic crust, communities of tube worms, clams, and specialized fish thrived in total independence from sunlight. The primary producers in these ecosystems were not photosynthetic, but chemosynthetic bacteria, organisms that derived energy from the oxidation of hydrogen sulfide and other reduced chemicals rather than from solar radiation. Giant tube worms, Riftia pachyptila, grew up to 8 feet in length despite having no mouth, no gut, and no anus, sustaining themselves entirely through a symbiotic relationship with chemosynthetic bacteria housed in a specialized organ called a trophosome. The worms absorbed hydrogen sulfide through their crimson plumes and transported it via specialized hemoglobin molecules to the bacteria, which converted it into organic compounds that nourished both microbe and host. The temperatures at these vents could exceed 700 degrees Fahrenheit, where the superheated water met the near-freezing ambient seawater, creating thermal gradients so extreme that organisms had to navigate microclimates varying by hundreds of degrees across distances measured in inches. The discovery of hydrothermal vent communities revolutionized scientific understanding of life's potential and raised questions about its origins, for the chemical conditions at these vents, rich in energy and chemical reactivity, shielded from the vagaries of surface conditions, resembled hypothesized conditions on the early Earth when life first arose. If life could emerge and thrive in such environments, independent of sunlight, then the range of potentially habitable environments expanded dramatically, encompassing not just the surfaces of planets in the narrow habitable zones around stars, but the interiors of icy moons like Europa and Enceladus, where liquid oceans might churn beneath miles of ice, heated by tidal forces rather than stellar radiation. The proteins that allowed vent organisms to function at extreme temperatures were thermostable variants of standard biochemical machinery, their amino acid sequences tweaked by millions of years of natural selection to maintain structural integrity at temperatures that would denature ordinary proteins into useless strands. Industrial applications followed the basic science, as these thermostable enzymes found use in everything from laundry detergent to the polymerase chain reaction that revolutionized molecular biology, allowing DNA to be amplified through cycles of heating and cooling that would destroy ordinary enzymes. Yet the deep ocean's extremes were not limited to the scorching environs of hydrothermal vents. The abyssal plains, those vast stretches of seafloor that constitute the largest habitat on Earth, presented challenges of a different sort. Here, at depths averaging about 13,000 feet, the temperature hovered just above freezing. The pressure exceeded 300 atmospheres, and food was so scarce that the benthos was essentially a desert, its sediment surface disturbed only by the occasional slow passage of a sea cucumber or the delicate tracks of a brittle star. The primary source of nutrition in this realm was marine snow, the constant rain of organic detritus that drifted down from the productive surface waters, dead phytoplankton, fecal pellets, molted exoskeletons, the occasional carcass of a larger animal. This material was so thoroughly consumed and recycled as it sank that only a small fraction of the organic matter produced in the sunlit zone ever reached the abyssal depths. And what did arrive was so depleted of nutritional value that abyssal organisms had evolved metabolisms of extreme frugality, burning energy at rates that made sloths seem hyperactive by comparison. Some abyssal fish had metabolic rates only a few percent those of their shallow water relatives, their muscles reduced to minimal mass, their bones thin and fragile, their movements languid and economical. The scarcity of food in the abyss meant that when a substantial foodfall occurred, the carcass of a whale or large fish sinking intact to the bottom, it represented an ecological event of enormous significance, an oasis of abundance in the desert of the deep. A single whale fall could sustain a complex succession of communities for decades, beginning with mobile scavengers like hagfish and sleeper sharks that consumed the soft tissues, progressing to polychaete worms and crustaceans that colonized the bones, and culminating in specialist bacteria that extracted lipids from the marrow itself. Chemosynthetic bacteria established themselves on whale bones, much as they did at hydrothermal vents, creating localized ecosystems that persisted for 50 years or more, islands of productivity on the otherwise barren plain. Certain species of bone-eating worms, discovered only in 2002 and appropriately named Osidax, bone devourer, had evolved the ability to penetrate whale bones with root-like structures and digest the lipids within, sustained by symbiotic bacteria housed in their tissues. These worms, like the giant tube worms of hydrothermal vents, lacked a digestive system of their own, having outsourced the biochemical work to their bacterial partners in what amounted to a microbial kitchen growing inside their bodies. The concept of symbiosis, the intimate association between organisms of different species for a mutual benefit, reached its most elaborate expressions in the marine realm, where the line between individual and colony, between self and other, often blurred beyond recognition. Corals, the architects of reefs that rivaled rainforests in their biodiversity, were not single organisms but chimeras, animals that had internalized photosynthetic algae called zooxanthellae within their tissues. The coral polyp, a simple creature related to jellyfish and sea anemones, provided the algae with a protected environment and access to sunlight, while the algae reciprocated by producing sugars through photosynthesis, providing up to 90% of the coral's energy needs. This partnership allowed corals to thrive in tropical waters that were paradoxically nutritionally impoverished. The water was clear precisely because it contained little plankton or other organic matter, and without their algal symbionts, the corals would starve. The calcium carbonate skeletons that corals secreted accumulated over geological time into structures visible from space, the Great Barrier Reef off Australia extending for over 1,300 miles, a monument to the collaborative achievements of animal and alga repeated trillions upon trillions of times across millennia. But coral reefs were not merely passive structures. They were dynamic ecosystems engaged in constant chemical warfare and negotiation. Corals competed for space by extending specialized tentacles at night to sting and digest adjacent colonies, engaging in slow-motion territorial battles that could last for years. They released chemical signals that attracted specific species of fish and crustaceans that, in turn, defested the coral against crown-of-thorns starfish and other predators, a protection racket millions of years in the making. The three-dimensional complexity of reef structures created niches for thousands of species, from tiny gobies barely an inch long that spent their entire lives within the branches of a single coral head, to groupers that could exceed 6 feet in length and weighed more than an adult human. Apex predators whose removal by overfishing could trigger cascading effects throughout the entire reef community. The nighttime reef revealed itself as a different world from its daytime incarnation as nocturnal species emerged from crevices and daytime residents retreated into shelter. Parrotfish secreting mucus cocoons that masked their scent from hunting moray eels that navigated by smell as much as by sight. The parrotfish themselves played an ecological role that extended far beyond their immediate feeding, for they were, quite literally, manufacturers of sand. Their beaks, fused teeth designed for scraping algae from coral rock, were among the hardest biological materials known, and in the course of feeding, parrotfish inadvertently consumed substantial quantities of coral skeleton along with their intended algal prey. This coral calcium carbonate passed through their digestive systems to be excreted as fine sand, each large parrotfish producing hundreds of pounds of sand annually. The white sand beaches of tropical islands were thus, in substantial measure, parrotfish excrement, a fact that might diminish the romance of a tropical vacation for some, but which illustrated the interconnectedness of marine ecosystems in ways that were both humbling and profound. The sand itself provided habitat for yet another suite of organisms, from burrowing clams to garden eels that swayed in the current like underwater wheat, their bodies anchored in individual burrows from which they plucked zooplankton carried past by the tide. The tidal rhythms that governed life in the shallows were driven by the gravitational interaction between Earth, Moon, and Sun, a cosmic clockwork that created currents and vertical migrations of water that organisms had evolved to exploit with remarkable precision. In temperate waters, the intertidal zone, that region between high and low tide marks, presented one of the most stressful environments on the planet, alternately submerged and exposed, its residents subjected to crashing waves, desiccating sun, and predation from both marine and terrestrial animals. Barnacles that encrusted the rocks in the high intertidal zone spent the majority of their lives exposed to air, sealed within their calcium carbonate plates, waiting for the brief hours of high tide when they could extend their feathery legs to filter feed from the water. They were crustaceans, relatives of crabs and shrimp, but so modified by their sessile lifestyle that early naturalists classified them as mollusks, only recognizing their true nature when their planktonic larvae were examined under the microscope. Charles Darwin, during the decades between his return from the voyage of the Beagle and the publication of On the Origin of Species, devoted 8 years to the study of barnacles, producing 4 monographs that established him as the world's expert on cirripedes and giving him the detailed comparative anatomy necessary to formulate his theories of evolution by natural selection. The evolutionary innovations that allowed marine organisms to colonize every available niche in the ocean included sensory systems so alien to terrestrial experience that they seemed almost supernatural in their capabilities. Sharks, apex predators that had patrolled the oceans for 400 million years, possessed an electroreceptive sense that allowed them to detect the weak electrical fields generated by the muscle contractions and nerve impulses of other animals. These electroreceptors, called ampullae of Lorenzini, were gel-filled canals opening to the surface through pores concentrated around the shark's snout, sensitive enough to detect electrical potentials as small as 5 billionths of a volt, roughly equivalent to the field produced by a battery with its terminals separated by 1,000 miles of seawater. This sense allowed sharks to hunt prey buried in sand, invisible to conventional vision, and to navigate using the Earth's magnetic field, detecting the electrical currents induced as they swam through the planet's geomagnetic lines. Great white sharks migrated thousands of miles between coastal hunting grounds and oceanic feeding areas, navigating with a precision that suggested an internal map of astonishing sophistication, returning to the same seal colonies year after year along routes that passed through featureless, open ocean where visual landmarks were non-existent. The great white's reputation as a mindless killing machine perpetuated by popular culture and sensationalist media bore little relation to the reality of an animal that exhibited complex behaviors suggesting considerable cognitive capacity. White sharks breached, launched themselves completely out of the water when hunting seals, an energetically expensive tactic that suggested calculated strategy rather than mere instinct. They approached prey from below and behind, using the sun's position and the background light to maximize their camouflage until the final explosive rush. They were also surprisingly cautious, often making initial exploratory bites on unfamiliar prey before committing to a full attack, which explained why human victims of shark bites often survived. The shark, having determined that the strange creature in the neoprene wetsuit was not the expected blubbery seal, often declined to pursue the attack further. Fatal shark attacks, while tragic, were statistically rare to the point of insignificance. Humans were far more likely to be killed by lightning, bee stings, or domestic cattle than by sharks. Yet the primal fear of being eaten by an unseen predator from below ensured that sharks occupied an outsized space in the human imagination. That imagination, fed by centuries of sailors' tales and fragmentary encounters, had created a bestiary of sea monsters that mingled fact and fiction in proportions that even experts struggled to untangle. The sea serpent, reported from cultures around the world and across historical eras, almost certainly represented multiple phenomena collapsed into a single category. Schools of dolphins swimming in line astern that at distance resembled a single undulating creature. Or fish, ribbon-like denizens of the deep that could exceed 50 feet in length and occasionally surfaced when sick or dying. Giant kelp torn loose from the seafloor and floating with its long fronds trailing like a serpent's body, and probably some sightings of genuinely large sharks or whales, seen under poor conditions that allowed the observer's expectations to shape interpretation. The oarfish, when finally captured and described scientifically, proved to be a creature almost as strange as fiction. Its body compressed laterally into a ribbon typically only a foot wide but potentially 50 feet long, crowned with a spectacular red crest that could be erected when the fish was alarmed. It swam by, undulating its extraordinarily long dorsal fin while holding its body nearly vertical, an energy-efficient mode of locomotion for an animal that drifted through the mid-water depths feeding on krill and small fish. Oarfish were likely responsible for many sea serpent sightings, their huge eyes and horse-like face matching descriptions from ancient mariners, and their habit of surfacing before earthquakes, possibly driven to the surface by changes in subsurface pressure or electromagnetic fields associated with tectonic activity, had given them a reputation as harbingers of disaster in Japanese folklore. The Japanese relationship with marine life, shaped by geography and necessity, had produced a culture of ocean observation more nuanced and detailed than perhaps any other pre-industrial society. In the Edo period, when Japan sealed itself from most foreign contact, naturalists produced elaborate illustrated encyclopedias of marine life that combined artistic beauty with genuine scientific observation. They documented the seasonal migrations of fish stocks, the relationships between currents and catches, and the life cycles of commercially important species with an attention to detail driven by practical necessity. Japan's mountainous terrain left little arable land, making the surrounding seas critical sources of protein. The Japanese had named and categorized dozens of species of jellyfish, recognizing distinctions that Western science would not describe for another century, and their fishermen had developed sustainable harvesting practices born from understanding that overexploitation would lead to collapse. The concept of satoyami extended to marine environments as satumi, recognized that humans were part of the ecosystem rather than separate from it, and that careful management could maintain productivity indefinitely, a philosophy that stood in stark contrast to the extractive approach that characterized much Western fishing. Jellyfish themselves, often dismissed as simple brainless blobs, represented an ancient and remarkably successful body plan that had persisted for at least 600 million years with only minor variations. Their bodies, composed of up to 95% water, were nearly neutrally buoyant, requiring minimal energy to maintain position in the water column, and their simple construction allowed them to grow to enormous sizes on minimal food input. The lion's mane jellyfish, found in cold northern waters, produced a bell that could exceed 6 feet in diameter, trailing tentacles over 100 feet long, creating a curtain of stinging nematocysts that paralyzed any small fish or crustacean unfortunate enough to blunder into it. These nematocysts, microscopic capsules containing coiled threads armed with barbs and venom, were among the fastest cellular processes known to biology, discharging in less than a millisecond with accelerations exceeding 5 million gravities, firing their hollow threads into prey with enough force to penetrate crustacean shells and inject venom directly into the victim's tissues. The venom itself varied by species from mild irritant to potently deadly. The box jellyfish of Australian waters carried enough venom to kill dozens of adult humans, and its sting caused pain so intense that victims often went into shock before the venom's cardiac effects could take hold. Yet even the deadly box jellyfish was a marvel of evolution, possessing a sophistication that seemed incongruous with its phylogenetic position among the most primitive multicellular animals. Box jellyfish had true eyes, 24 of them, arranged in clusters around the bell, complete with lenses, retinas, and visual pigments similar to those found in vertebrates, though their evolutionary path to image-forming eyes was entirely independent, a striking example of convergent evolution. These eyes allowed box jellyfish to navigate through mangrove channels and avoid obstacles, swimming with the directional control that other jellyfish, which drifted largely at the mercy of currents, could not match. The presence of complex eyes in an animal with no centralized brain raised profound questions about the nature of vision and information processing. Where exactly did the visual information go, and how was it processed in the distributed nerve net that constituted the jellyfish's nervous system? Recent research suggested that some degree of image processing occurred within the eye itself, the neural tissue of the retina performing computations that in more complex animals would require substantial brain tissue. An example of distributed processing that engineers studying parallel computation found intriguing. The transparent bodies of jellyfish and many other pelagic creatures represented an adaptation for camouflage in the open ocean, where there was no substrate to hide behind and no background to blend against. Transparency eliminated shadows and made the animals nearly invisible. Though perfect transparency was difficult to achieve, the digestive system typically remained visible as a dark mass, and any pigmentation for light sensing or other purposes created visible spots. Some organisms solved this problem through counter-illumination, producing light from their ventral surfaces that matched the intensity and wavelength of sunlight filtering from above, effectively erasing their silhouette when viewed from below. The hatchet fish, a small midwater species with a body literally shaped like a hatchet blade, carried photophores along its belly that it adjusted constantly as it migrated vertically, maintaining perfect camouflage across 1,000-foot depth range as the ambient light levels change from relatively bright to vanishingly dim. This required sophisticated light sensing and a control system that could adjust the intensity of dozens of individual photophores independently. Biological technology that functioned with a precision that seemed improbable in a fish the size of a human thumb. The vertical migrations that hatchetfish and countless other species undertook daily represented the largest movement of biomass on the planet, a phenomenon invisible to human eyes but detectable by sonar as a deep scattering layer that rose toward the surface each night and sank to depth with the dawn. Billions of tons of fish, squid, krill and other organisms migrated vertically through hundreds or even thousands of feet of water column, feeding in the productive surface waters under cover of darkness and retreating to depth during daylight to avoid visual predators. This migration transported nutrients and carbon from surface to depth, playing a role in the ocean's biological pump that sequestered atmospheric carbon dioxide into the deep ocean, where it could remain isolated from the atmosphere for centuries or millennia. The scale of this migration was so vast that it affected oceanographic properties. The warm bodies of countless animals creating measurable temperature anomalies, their metabolic activity consuming oxygen and producing carbon dioxide in quantities sufficient to alter the chemical properties of the water masses they inhabited. Among the migrants were larval forms of organisms that would eventually settle to the bottom or attach to reefs, their planktonic phase serving as a dispersal mechanism that allowed species with sessile or relatively sedentary adult forms to colonize new habitats and maintain genetic connectivity across populations separated by hundreds or thousands of miles. The larval phase represented a distinct life stage with its own morphology, behaviors, and ecological niche, often bearing so little resemblance to the adult that early naturalists classified them as separate species entirely. The larvae of spiny lobsters were transparent, flattened creatures that drifted in the plankton for months, bearing no obvious resemblance to the heavily armoured crustaceans they would become. Sea urchin larvae were tiny, bilaterally symmetrical organisms that swam using bands of cilia. Their internal organs are visible through transparent tissues, revealing no hint of the radial symmetry that would characterize their adult form. The metamorphosis from larva to adult required wholesale reorganization of body structure, certain tissues dissolving and being reabsorbed while new structures formed from clusters of undifferentiated cells called imaginal discs, a transformation as profound as any in biology, comparable to the metamorphosis of caterpillar to butterfly, but occurring entirely underwater and largely unstudied except by specialists. The specialists who did study marine larvae were part of a scientific tradition that stretched back to the 18th century, when microscopy first revealed the previously invisible worlds of the very small. Anton van Leeuwenhoek, the Dutch draper who ground his own lenses and became the first person to observe bacteria and protozoa, also examined seawater samples, documenting the remarkable diversity of microscopic life suspended in every drop. His observations, communicated in letters to the Royal Society written in colloquial Dutch rather than scientific Latin, described rotifers, copepods, and dinoflagellates with an enthusiasm unmarred by formal education, but informed by meticulous observation and genuine curiosity. The copepods that Leeuwenhoek observed were the dominant herbivores in the ocean, arguably the most numerous multicellular animals on Earth, with population estimates in the billions of trillions. Individual copepods were small, rarely exceeding a few millimeters, but their collective grazing pressure shaped the composition of phytoplankton communities and their fecal pellets constituted a significant fraction of the marine snow that fed the deep ocean. Some copepod species exhibited bizarre sexual dimorphism, the females carrying paired egg sacs like saddlebags, while males remained permanently attached to their mates as tiny parasitic appendages. Another example of the ocean's tendency toward extreme reproductive strategies. The dinoflagellates that shared the water with copepods included species capable of bioluminescence, creating the sparkling trails and glowing waves that had enchanted mariners for millennia and inspired both poetry and superstition. When present in sufficient numbers, disturbed dinoflagellates would flash blue-green light in response to mechanical stimulation, creating ghostly illumination around ships' bows and swimming animals, a phenomenon that puzzled natural philosophers until the development of chemical theory allowed the luminescent reaction to be understood as a specialized version of ordinary oxidative metabolism. Some dinoflagellate species were also responsible for harmful algal blooms, the red tides that could poison fish and shellfish and cause respiratory irritation in humans living along affected coasts. These blooms occurred when conditions of temperature, nutrients, and water stability aligned to favour explosive dinoflagellate reproduction, populations doubling every few days until the water became thick with cells and discoloured by their pigments. The toxins that some species produced served no clear function for the dinoflagellates themselves, appearing to be byproducts of cellular metabolism rather than defensive chemicals, yet they were among the most potent neurotoxins known to science. Saxitoxin, produced by certain dinoflagellates and concentrated up the food chain in filter-feeding shellfish, blocked sodium channels in nerve cells with such efficiency that a few milligrams could kill an adult human, and there existed no effective antidote. The phenomenon of biomagnification, whereby toxic substances became concentrated at each step up the food chain, affected not only naturally produced toxins, but also the synthetic chemicals that industrial civilization had released into the marine environment. Heavy metals like mercury, organochlorin pesticides like DDT, and industrial compounds like PCBs were lipophilic, accumulating in fatty tissues rather than being excreted, such that top predators like tuna, swordfish and sharks carried concentrations thousands or millions of times higher than the background levels in seawater. Marine mammals were particularly vulnerable, their blubber serving as both insulation and as a reservoir for accumulated pollutants that could be mobilized during periods of fasting or lactation, poisoning adults and being transferred to nursing young in milk that contained toxin concentrations high enough to compromise immune function and reproductive health. The belugas of the St. Lawrence River estuary, living in waters contaminated by decades of industrial discharge, carried body burdens of pollutants so high that when they died and washed ashore, their carcasses legally qualified as hazardous waste requiring special disposal procedures. The resilience of marine ecosystems in the face of human impacts varied tremendously, depending on the specific communities and the nature of the disturbance, with some systems proving remarkably robust, while others collapsed with shocking rapidity once critical thresholds were crossed. Kelp forests, those towering underwater groves of brown macroalgae that could grow 2 feet per day in ideal conditions, created three-dimensional habitats along temperate coastlines that supported hundreds of species from tiny bryozoans to sea otters and harbour seals. The kelp themselves, despite their plant-like appearance, were not true plants, but rather members of the kingdom Protista, more closely related to the dinoflagellators and diatoms than to terrestrial vegetation, their evolutionary path to large multicellular forms entirely independent of the one that produced trees and flowering plants. They anchored to rocky substrate with a holdfast, a root-like structure that served only for attachment rather than nutrient absorption, all of their nutrients being absorbed directly from seawater through their blade surfaces. This mode of nutrition made them sensitive to water quality, turbidity reducing the light available for photosynthesis, and smothering by sediment blocking the molecular exchange of nutrients and gases through their surface tissues. The cascade of ecological effects that could follow the removal of a single species from kelp forest ecosystems illustrated the complexity of marine food webs and the importance of species whose impact on the immunity far exceeded their numerical abundance. Sea otters, hunted nearly to extinction for their luxurious fur in the 18th and 19th centuries, were a keystone species in kelp forests, their predation on sea urchins preventing the urchins from overgrazing the kelp. Where otters were absent, urchin populations exploded, their constant grazing reducing the kelp forest to urchin barrens, rocky substrates covered in purple or green urchins, with only stubble remaining of what had been towering algal canopies. The loss of the kelp meant loss of habitat for fish, invertebrates, and the entire associated community. A phase shift from complex productive ecosystem to impoverished wasteland triggered by the removal of a single predator. The reintroduction of sea otters to parts of their former range allowed kelp forests to recover, the urchin populations crashing once their primary predator returned, the kelp regrowing, and the associated species recolonizing from refugia or adjacent areas. Though the recovery took decades and required that viable otter populations establish themselves and remain protected from poaching and other threats. Sea otters were unusual among marine mammals in lacking the thick blubber layer that insulated whales, seals, and sea lions from frigid waters, instead relying on the densest fur in the animal kingdom, up to 1 million hairs per square inch, to trap air and provide thermal insulation. This required the otters to groom constantly, maintaining the air-trapping structure of their fur and removing any oil or debris that would compromise its insulating properties. The energetic cost of this grooming, combined with the metabolic demands of maintaining body temperature in water that conducted heat away 25 times faster than air, meant that sea otters had to consume roughly 1/4 of their body weight in food daily, spending the majority of their waking hours either foraging or eating. Their tool use, employing rocks to crack open shellfish, carrying favourite hammer stones tucked into armpit pouches, and sometimes using kelp fronds to anchor themselves while sleeping, demonstrated cognitive capabilities and behavioural flexibility, rare among non-primate mammals, and populations exhibited cultural transmission of foraging techniques, different communities specialising in different prey species and employing distinct methods passed from mothers to pups across generations. The concept of culture, of learned behaviours transmitted socially rather than genetically, had been documented in cetaceans as well, most extensively studied in orcas, whose populations exhibited distinct ecotypes with different prey preferences, hunting techniques, and vocal dialects that were maintained even when different ecotypes occupied overlapping ranges and must surely have encountered one another. Resident orcas in the Pacific Northwest specialized in salmon, hunting cooperatively and using echolocation to track fish schools in turbid waters, while transient orcas hunted marine mammals, approaching stealthily without echolocating, presumably to avoid alerting their acoustically sensitive prey, and employing sophisticated coordinated tactics to overcome animals as large as grey whales. These behavioural differences were not hardwired genetically, but rather culturally transmitted. Young orcas learning from their mothers and other pod members through years of observation and practice, gradually acquiring the specialised knowledge necessary to hunt effectively in their particular ecological niche. The vocal dialects that distinguished different orca populations were similarly learned, each pod having a repertoire of discrete calls that its members used in specific contexts, the calls varying enough between pods that researchers could identify individuals by their vocal signatures, much as human languages varied between communities and allowed listeners to identify a speaker's origins. The intelligence required to navigate complex social dynamics, coordinate hunts involving multiple individuals, each playing specialized roles, and transmit cultural knowledge across generations suggested that orcas and other large-brained cetaceans possessed cognitive capacities rivaling or potentially exceeding those of great apes, despite having brains organized along fundamentally different architectural principles reflecting their independent evolutionary history. The neocortex of a dolphin or whale was highly convoluted, with more surface area relative to volume than even human brains, though the cortex was thinner, comprising fewer cellular layers. The distribution of different cell types differed from terrestrial mammals, and the functional mapping of the brains remained poorly understood compared to the extensively studied primate brain. But the behavioral evidence of complex cognition was undeniable. Dolphins had been observed using marine sponges as tools, breaking them off the seafloor and wearing them over their rostrums while foraging in rough substrate, protecting their sensitive snouts from abrasion in what appeared to be a learned behavior transmitted matrilineally in certain families. They engaged in cooperative foraging strategies, sometimes working with human fishermen in traditional partnerships that benefited both species, the dolphins herding fish toward nets in exchange for a share of the catch. Relationships that in some communities had persisted for generations, with knowledge of the partnership passed down on both sides. The capacity for learning and behavioral flexibility became increasingly important as marine organisms confronted environmental changes occurring at rates unprecedented in recent geological history, the ocean absorbing not only industrial pollutants, but also excess heat and carbon dioxide from the burning of fossil fuels, altering the fundamental chemistry and physics of the marine environment. The ocean had absorbed over 90% of the excess heat trapped by greenhouse gases, buffering the atmosphere from even more severe warming, but at the cost of thermal stress to marine organisms adapted to relatively stable temperatures. Coral bleaching, the expulsion of symbiotic zooxanthellae that gave corals their colour and provided most of their nutrition, occurred when temperatures rose even a degree or two above seasonal norms for periods of weeks, the stressed corals ejecting their symbionts in a response that could be fatal if conditions did not improve quickly enough for recolonization. Mass bleaching events had become increasingly frequent and severe, with some reef systems experiencing near-total mortality across hundreds of miles, raising questions about whether these ecosystems could persist in their current form, or whether they would transform into fundamentally different assemblages dominated by more temperature-tolerant but less diverse organisms. The absorption of atmospheric carbon dioxide into seawater produced carbonic acid, gradually lowering the ocean's pH in a process termed ocean acidification that threatened organisms relying on calcium carbonate for their shells and skeletons. As seawater became more acidic, it became more difficult for these organisms to extract carbonate ions from solution to build their protective structures, and if pH dropped sufficiently, existing calcium carbonate structures would begin to dissolve, a prospect particularly alarming for corals, mollusks, and planktonic organisms like pteropods. Tiny swimming snails whose delicate shells were already showing signs of dissolution in high-latitude waters where cold temperatures and upwelling deep water had made surface waters more acidic than in tropical regions. The pteropods formed a crucial link in polar food webs, constituting a major food source for fish, seabirds, and baleen whales, and their decline could trigger cascading effects throughout the entire ecosystem, demonstrating once again that the fate of charismatic megafauna was ultimately tied to the health of microscopic organisms that few people outside the scientific community had ever heard of or seen.